I would have two announcements before the next talk. The first announcement is about the parallel session speakers. Kindly hand over your presentations so that it can be loaded in the computer. Then tea is ready outside, and whoever is interested can take it towards the right of the stage, please. The next session will continue.
So is Dr. Chandak around? Where is he?
Idhar sir, idhar.
अच्छा तो वो अंदर आ गया ठीक है मुझे दिखे नहीं है.
You wanted me to present from audience.
Every day you are getting younger. So the talk is very simple and one can remember the title DoHAD and risk of non-communicable diseases, and Dr. Chandak is a very well-known basically clinical research scientist from our own adjacent CCMB, and I welcome you to give your talk, Girish.
On karna. Abhi on hai. Okay.
So Dr. Giriraj, you have 20 minutes to talk and yes, 5 minutes to interrupt.
You can stop me whenever the time is done.
I'll remind you before 2 minutes.
Yes, that should be fine. Okay. So, thank you very much, and thanks to the organizers and specifically to Uma. I think it's because of his perseverance that I am able to be here, and my students will not spare you because they had to work very, very hard for making me able to speak over here. So, friends, today I will speak about a concept which is called developmental origin of health and disease. My lab has been working in this area for last 2 decades, and essentially we have some interesting observations which we think are useful to prevent the risk of non-communicable diseases. So, before I begin, I would like to say that the work that I am presenting has elements from lots of people in my lab who have worked with me for last several years, and I will illustrate that how this whole picture is justified. Thanks to my collaborators, I have different cohorts from different parts of the country which allow me to understand the observations and spread them to the width and breadth of this particular country. Also, we have international collaboration which also allows us to compare both the genetics as well as the nutritional components against the European people as well. So, these are my collaborators. Each of them have been the pioneer in generating various cohorts, the benefits of which I have utilized. So, before I begin, there are 2 terms in my title which I would like to explain because they form the basis of the whole presentation. First of all, the non-communicable diseases and associated traits. I don't need to tell this audience that type 2 diabetes or hypertension or cardiovascular problems, they are the non-communicable diseases. What is important to understand is that there are both genetic and environmental factors. Just to give an example as simple as human height itself, there are so many things which can actually influence the mother's height, the father's height, which are largely through genetics, but there are many other factors, the maternal factors as well as the child factors which can influence the final level of height, and height is known to be associated with risk of cardiovascular problem. This is what is the link between non-communicable diseases, a trait which is associated with that, and the component of genetics as well as the environmental factors. The DoHAD essentially tries or rather attempts to connect both the genetics as well as the environmental aspect. And in terms of environmental aspect, it talks about specifically the intrauterine or the early life environment. There is enough evidence which has been provided by Professor David Barker, who is the pioneer of this hypothesis, which indicates that very early in life the determination of the disease risk for non-communicable diseases is established. It is almost 35 years back that this hypothesis was advanced, and lot of work has happened. I was fortunate to be in this arena quite close to the time then when it started, and that is why we have been able to make some seminal contribution, particularly from low and middle income countries like India. So, to summarize this slide, that according to Barker or this DoHAD hypothesis, it is the maternal nutrition which determines the fetal nutrition and obviously its growth, and this determines the birth size which decides whether a baby is going to be healthy or tall or whatever, and we know that this particular development is the risk factor for disease, not particularly non-communicable diseases. So, overall, to say that fetal programming is the common word of DoHAD, which means that this in fact summarizes or leads to long latency disorders which are associated with intergenerational transmission. So, to fulfill this particular objective, I have been privileged to have been working with various cohorts. I would not be describing each of them, but suffice it to say that there are all these cohorts, they represent different parts of the country. These cohorts, many of them are starting even early, even before pregnancy itself, and then the follow-up data has been available right from pre-pregnancy up to the age of 80 years. And when I say the data is available, this means that at every 6 years the follow-up is there, and during pregnancy and during the different trimesters of pregnancy, various data sets and biological samples are available. As you can see here, these are the common outcomes which have been investigated in this cohort, and this is under the banner of SNEHA, which is Society for the Natal Effects on Health of Adults. And we have been able to work together for close to last 2 and 2.5 decades, and I just want to give a glimpse of what we have been able to do in last few years, and then I will come to the recent exciting story which is what probably will keep you awake and interested. So to give you an example of a cohort, how does this cohort look like? This is Mysore Parthenon cohort. This was established as a longitudinal cohort, wanted to investigate that how the maternal nutrition during pregnancy influences offspring's future health and risk of cardiometabolic disorders. And as you can see that the women were recruited during pregnancy, and these are the follow-up stages. These are the various parameters which include all biological biochemical parameters as well as various anthropometric, glycemic or cardiometabolic parameters. To summarize that, what we observed from many of these cohorts, this was one of the first cohorts where we found that actually vitamin B12 deficiency is present in close to 40% of the individuals, which was a big serendipitous finding because B12 has a very low requirement or very low RDA, but to find that almost half of the population is B12 deficient really raised the alarms. Subsequently, through Pune maternal nutrition study, which is run by Dr. Yagnik, we could show that actually it is not only B12, but the folate B12 imbalance which influences the future possibilities of insulin resistance and future possibilities of cardiometabolic diseases. We showed that those who have mothers who have high vitamin B12 levels, their kids were more obese at 6 years. Those mothers who had high folate levels, their kids at 6 years were more obese. And finally, when we combine the B12 and folate, because that is a part of the 1 carbon metabolism cycle, we find that mothers who had lowest level of B12, highest level of folate had the kids who were most insulin resistant. So, the proof of the pudding is in eating, and that is what this slide demonstrates, that if there is a folate B12 imbalance in the mothers, then their kids are born with a risk of developing insulin resistance as early as 6 years. I may just mention that in a recent follow-up at the age of 24 years, we find that close to 28% of these children are already pre-diabetic. To further extend, we also showed that B12 levels are also influencing the occurrence of gestational diabetes, which occurs during the pregnancy. And as you would see that if there were B12 deficient mothers during pregnancy, they had kids, they themselves were more obese, and if they continued to be deficient, then they were more likely to persist with GDM. And if you see them 5 years later with persistent B12 deficiency, they continue to remain diabetic as well. And then its effect on the children in the next generation, as I mentioned with the DoHAD hypothesis, that if there are offsprings born to diabetic mothers or offsprings born to diabetic fathers, they clearly have an altered subscapular skin fold, which is a measure of central obesity, and also other parameters even as early as 13 to 14 years of age. These slides essentially demonstrate that there are early life exposures. Please consider B12, folate and others as a surrogate of any early life exposure, but we demonstrate that B12, folate and other early life exposures can program the fetus, and the phenotype of risk of cardiometabolic disorders or other non-communicable diseases can arise as early as adolescence. What was interesting was that it was the problem that we identified, that okay, B12 does this, but what is the solution for that? And what we found that in India it's a major problem. At all ages, in both genders, the B12 levels are relatively lower compared to Europeans. And then we hypothesized that probably this particular exposure could be one of the mechanisms by which we are having this global epidemic of diabetes and cardiovascular problems, and which gives us the bad name of being the diabetic capital of the world. What we did was that because homocysteine is the ultimate metabolite which is in the 1-carbon metabolism, we looked at the B12 supplementation and showed that B12 supplementation reduces homocysteine level, which provided us the proof that if there are any mechanisms or any alterations which are occurring because of B12 deficiency or because of raised homocysteine level, then by B12 supplementation at an appropriate stage, those effects are likely to be reversed. And suffice it to say that in this huge study, which was on 6,000 women, where we supplemented these women during their pregnancy with a food-based diet which had multiple micronutrients including B12, we showed, we showed that these are for the all women and this is the birth weight. We showed that the women in the B12 supplemented group had kids which had higher birth weight, and particularly this interacted with the maternal BMI, which means that mothers with better BMI or better health status would have kids who will have better birth weight, better body composition, and probably a lesser risk of non-communicable diseases. Subsequently, we also showed that in this B12 related deficiency mothers, the baby's phenotype is very much associated with the cognition as well, which means that overall any altered exposure during the intrauterine life to the fetus can have an influence on the child's phenotype. What could be the mechanism? Just to show this 1-carbon metabolism cycle without going into much detail, suffice it to say that if there is an alteration between, if there are low B12 levels, it sits at the connect between folate and methionine, and in fact that means that the SAM production will be reduced and there is a possibility that the methylation potential of the cell will be altered. We provided the evidence of this by doing a B12 supplementation, and we showed that specifically the B12 supplementation did a differential methylation of a microRNA MIR21, and it also had the differential methylation of FTO, TCF7L2, which are the genes associated with type 2 diabetes. And thus we provided the evidence that whether the genetic susceptibility to type 2 diabetes is because of several genes, but then environment, specifically in terms of B12, can also influence the methylation and in turn have a risk of development of type 2 diabetes. So, overall what we have been able to provide with these cohorts is the evidence of both nutrient mediated and fuel mediated teratogenesis, and that is a problem in India because Indians have both undernutrition as well as overnutrition. And this is what through these cohorts we have been able to show that the nutrient like B12 and others and glucose which are related with gestational diabetes, they both are linked to this particular cycle. Now, I told you a big story about various exposures, various phenotypes, various outcomes, but then it's not, body is not going to evolve in such complex pathways. We argued that there could be certain specific common mechanisms by which this pathway might be altered. And sorry. So, DoHAD hypothesis suggests that if there is any nutritional insult during critical periods of development, there can be influence of neuroendocrine structure as well as the function. And this way the early life exposures may lead to complex disease phenotype by common mechanisms such as an abnormal stress response. And we hypothesize that a deregulated HPA axis response and exaggerated stress reactivity can promote a programming of this kind of effect. Now, the work that I am going to present today is actually my privilege, but most of the work has been done by Dr. Kitty, Dr. Kumaran and Dr. Caroline. They have largely looked at the epidemiological responses, and the work in my lab has been done by Sohail, Sara and Swati, and I am having the privilege of being able to present in front of you. So, the first question that we asked was that does early life exposure influence the response to stress? And again we took Mysore Parthenon cohort, where as I mentioned earlier that there are these serial measures, serial samples and serial measures that are available with us. We specifically took 13.5 years time point, and what we did was we created a social stress in the participants. This was close to 262 kids who were born to these mothers. We had information on the maternal phenotype, as I already mentioned to you. What we did was we had this Trier social stress test, which means that all the participants after being called to the place, they are showed a calming video, and after that they are given random words by which they have to prepare a story in 10 minutes, and then they have to come and present in front of unknown people a story in 5 minutes and then some very basic maths task in 5 minutes, and then they are asked to wait in the waiting room. During this time their saliva is collected and also their cardiac responses are collected during that time continuously, and finally a calming video is shown to them. And these are the measurements which are the cardiovascular responses which have been looked at. So, this is the way this test is conducted, and the prospect of social evaluation by judges, it triggers a stress response, and this is a standard model that has been used in various studies, and frequent saliva samples and cardiac monitoring was being done. So, these are the results from that study which had about 259 participants which we analyzed. What we found that the kids who were born to mothers with low B12 level, they had a higher cortisol response. Similarly, as I mentioned that the B12 deficiency and high homocysteine concentration are each other's consequence. So, high homocysteine concentration specifically had a similar effect, and they also showed the higher cortisol and cardiovascular responses. As I mentioned that these are the tests which were looked at: the heart rate, the cardiac output, the stroke volume, the peripheral cardiovascular resistance, and as you would see that each of them had the effect because of high homocysteine concentration, specifically heart rate and cardiac output were statistically significant. Interestingly, we didn't find any association with folate deficiency or any gender interaction. Gender interaction specifically because this was in the adolescent stage and we wanted to understand specifically in the background of our animal studies which had shown some gender specific effect in these kind of influences. So, to summarize this slide, the early life exposure to altered 1-carbon metabolism as reflected by low B12 level programs the stress response as early as in adulthood. As I mentioned that we also had the follow-up samples at 21 years. So, understand that whether this is really a programming or it is a chance event, we further investigated or asked the same question that what is the status of altered stress response to early life exposure in 21-year-old children. And so, we 2 minutes, yeah. Okay. So, what we showed was that, so this is a little busy slide, but you have to believe me what I say is that what we found that in the offspring who were born to mothers with low B12 levels, they had a high baseline cortisol which was largely driven by females. Overall, the offspring of these mothers had low B12 level during pregnancy and this is what was the association. But what we found that there was a difference between genders, females and with males. However, the cardiovascular responses, they showed similar trend as in adults. Interestingly, we also found that the altered folate and homocysteine also had an influence on the various cardiovascular responses. As you would see that both heart rate and cardiac output responses are also altered. So, we summarize that the early life exposure to altered 1-carbon metabolism programs the stress response in adolescents which persists in young adulthood and has cardiovascular consequences. We wanted to understand what is the molecular basis of this altered stress responses. And to summarize again that we understand that the HPA axis and cortisol pathway is responsible for the cortisol level. There are two specific genes which I will talk about, the NR3C1 which includes cortisol receptor and 11 beta HSD2 which encodes the enzyme that inactivates cortisol. We hypothesize that the programming could be because of an altered methylation of these two genes which are playing critical role in the steroid metabolism. What we found that at 21 years, the methylation in NR3C1 and 11 beta HSD1 was associated with baseline cortisol and cortisol response, but that was largely driven by females. Whereas, when we look at the cardiac response, we found that the methylation of both of these genes was influencing the stroke volume. The higher methylation of these was associated with higher baseline cortisol and lower stroke volume response. Further, we investigated that whether the early life exposures have an association with the DNA methylation at 21 years. Again, we find that there is an altered methylation which is showing gender specific effect. So, to summarize this particular thing, that if we found that the folate and hyperhomocysteinemia, they had increased methylation of NR3C1 and had an association with baseline cortisol response and this cardiometabolic. Similarly, B12 concentration and homocysteinemia, they had influence on the HSD11 beta 2 methylation. So, to conclude, what we have shown is that the altered 1-carbon metabolism during pregnancy programs the cortisol response and consequent response to cardiovascular traits in early adolescence, which persists in the adult life. Although the nature of cortisol response was reversed, in contrast to the 13.5 years where it was higher, in 21 years it was lower, but the cardiac consequences persist and reflect the persistence influence of altered cortisol metabolism. As I mentioned that there was an increase in the baseline cortisol level, which indicates that the early life programming at 13.5 years had an influence on the stress response, but then over a period of time this became persistent with a persistently high levels of baseline cortisol, and that is why the cardiac consequences were continuous there. Fetal programming shows gender-specific association, possibly due to interaction of sex hormone around and post-puberty. Baseline cortisol in adults are strongly associated with the methylation in these two, which shows significant association with altered 1-carbon metabolism. We think that a persistence of abnormal cortisol response and differential methylation in the promoter region of these two genes are likely to have a causal role. This is the model that my student has prepared, which is the summary of the whole work. So, we have already an evidence of disturbed maternal 1-carbon metabolism during pregnancy, and we showed that there is association with altered cortisol level and stress response both in adolescence and adulthood, which is also responsible or associated with adverse cardiovascular response to stress. And what we possibly think is that this altered promoter methylation could be a programming mechanism which influences the expression levels of NR3C1 and 11 beta HSD2 and influences the circulating cortisol level. I must admit that, I would like to say that it's very simple to look at individual genes, but we think that it's an interaction between 11 beta HSD2 and the NR3C1 regulation which finally decides the cortisol response and subsequently the risk of non-communicable diseases. I will skip this. So, the last slide is that in non-communicable diseases, the usual dogma is that there is genetic susceptibility which is influenced by the precipitating factors like obesity, diet and others, but stress could be one of the very common factor which might link all of these, and that is what is DoHAD programming's contribution in the risk of non-communicable diseases. So, thank you very much, and I am sorry I took a little more time this, but
thanks,
happy
Dr.
to take any questions.
Thank you, Dr. Chandak. 2 questions at the most, time less. Both of you, that's it. Short question, short answer.
Maternal?
Mic का आवाज बढ़ा दो यार.
Mic.
Increase the sound. There is no sound in the mic.
Fostering, yeah. Yes. So I must say that I just presented this as a B12 and other exposure because that's the continuous story. But we have data on maternal socioeconomic status, fostering, and then early life adverse influences on the child, and each of them we are investigating independently. The fostering and others, they have an influence on the cognitive responses. So, each of the exposure is not likely to go to the same phenotype, but suffice it to say that these early life exposures have an influence on these phenotypes associated with NCDs.
Modi.
Yeah. Hi. Thanks.
I was curious, the very nice thing which you showed was sex-specific differences in the responses to vitamin deficiency.
Yeah.
What could be the mechanism? I didn't see that in your epigenetic
Right. So, actually this is the question in our mind also. So, this dates back from even our rat experiments also, where we generated B12 folate model, and we found that in the next generation there are gender specific effects. In fact, Shantanu, he did proteomic analysis and showed specifically that there are altered levels of sex hormones, and there are different kind of interactions. We also assume that it is the case. There is a lacunae over here because the first evidence which we have seen is in 13.5 years. We think that during that time the hormonal disturbances and others may not have allowed this phenomenon to be seen by us. Now what we want to do is that we want to look at the methylation in 13.5 years also and see that whether a gender specific changes in methylation could be there.
Okay, Chandak.
Sorry?
We have only peripheral blood and saliva, because peripheral blood and saliva we have, but we have looked at the correlation of this methylation in saliva with the peripheral blood as well, and there is a good concordance in that.
So Deepak,
It's unfortunate we can't have the human tissues.
so thanks, very good talk.
Thank
so
you.
do you think that Shantanu and Gupta says homocysteine is good? And
Shantanu Sen Gupta?
says homocysteine is good in adults and
No comments.
no comments.
No, homocysteine is not good for sure,
Okay.
but B12 is good, I feel.
Beautiful. So it's a wonderful talk, and let us give him a
Very
big
much.
clap. So in the meanwhile, we can invite in the meanwhile, we can invite the next speaker, Dr. A. Balasubramanian from Madras Diabetes Research Foundation, who will be speaking on emerging role of endocrine disruptors in diabetes as well as GDL.
I would like to request Professor Michael Murlidas to kindly hand over the memento to Dr. Chandak. The next talk of Dr. M. Balasubramanian will be an online talk, but he is ready to take the questions at the end. Another small announcement, please. I would like to request the session 5 orators to